I am Ritik Pier of first semester MSc Biochemistry. In this presentation, I am going to explain about PCR, which stands for Polymerization Chain Reaction. Introduction to PCR. PCR or Polymerization Chain Reaction is a technique used in molecular biology to create several copies of certain DNA segment. This technique was developed in 1983 by Carey Mullis, an American biochemist. PCR has made it possible to generate million copies of small segment of DNA. This tool is commonly used in molecular biology and biotechnology labs. Principle. The PCR technique is based on the enzymatic replication of DNA. In PCR, a short segment of DNA is amplified using primer-mediated enzymes. DNA polymerase synthesizes new strand of DNA complementary to the template DNA. The DNA polymerase can add nucleotide to the pre-existing 3' OH group only. Therefore, a primer is required. Thus, more nucleotides are added to the 3' end of the DNA polymerase. Components which are used in PCR technique are as follows. DNA template, the DNA of interest from the sample. DNA polymerase. Taq polymerase is used. It is a thermostable and do not denature at very high temperature. Oligonucleotide primers. These are the short stretch of single-stranded DNA complementary to the 3' end of sense and antisense strands. Deoxyribonucleotide triphosphate. These provide energy for polymerization and are the building blocks for the synthesis of DNA. These are single unit of bases. Buffer system. Magnesium and potassium provide optimum condition for DNA denaturation and renaturation. It is also important for fidelity, polymerase activity, and stability. These are the components which are required for PCR technique. Steps involved in PCR. The PCR involves three major cyclic reactions: denaturation, annealing, and elongation. Denaturation. Denaturation occurs when the reaction mixture is heated to 94 degrees Celsius for about 0.5 to 2 minutes. This breaks the hydrogen bond between the two strands of DNA and converts it into a single strand DNA. The single strand DNA now acts as a template for the production of new strand of DNA. The temperature should be provided for longer time to ensure the separation of two strands. After denaturation, annealing process takes place. In this process, the reaction mixture is lowered to 54 to 60 degrees Celsius around 20 to 40 seconds. Here, the primers bind to their complementary sequence on the template DNA. Primers are single-strand sequence of DNA or RNA around 20 to 30 base pair in length. They serve as starting point for the synthesis of DNA. The two separated strands run in opposite direction and consequently there are two primers. One is forward primer and another is reverse primer. Elongation. Elongation is the final step involved in polymerization chain reaction. At this step, the temperature is raised to 70 to 80 degrees Celsius. The bases are added to 3' end of the primer by the Taq polymerase enzyme. This elongates the DNA in the 5' to 3' direction. These three steps are repeated 20 to 40 times in order to obtain number of sequence of DNA of interest in very short period of time. Application of PCR. In medicine, testing of genetic disease mutation, monitoring the gene in gene therapy, in forensic science, used as a tool in genetic fingerprinting, identifying the criminals from millions of people, research and genetics. Compare the genome of two organisms in genetic study, analysis of gene expression, gene mapping. Thank you.